Number 6. Positive thigmotropism is a response in plants in which they move and grow toward an object the plant comes into physical contact with, usually curling around the object. This response of the shoot system benefits the plant by allowing the plant to produce more pollen for pollinators, take in more carbon dioxide to convert into glucose, absorb more water to transfer to the leaves, or reach more sunlight for photosynthesis. So the only thing different that we see here is the addition of a stick, and the tendrils of the plant are winding around the stick and the plant is growing upwards. And also in the question it mentions about physical contact. So that physical contact and it curling upwards should lead us to D, reaching more sunlight for photosynthesis. So there's nothing there that shows us roots or xylem, nothing about the stomata and carbon dioxide. And definitely you can't see anything there with the flowers and the pollen. So our best choice is D. Number 7. White flowers, Trifolium repens, are plants that are native to Europe and Central Asia and are able to produce the poison cyanide within their cells. Skunk cabbage, Symplocarpus foetidus, is a plant that is native to eastern North America and can produce a chemical that makes it have a strong, foul odor and bitter taste. The chemicals produced by both the white clovers and skunk cabbages are adaptations that allow them to attract more pollinators. Doesn't seem like there's anything going on there that would attract anything to them, maybe repel things from them. Survive along drought, nothing there about water. Protect against herbivores. Well, herbivores do eat plants, and these are plants. So it seems like this is something that plants wouldn't want to eat because they stink and they're gross, or they'll kill them, or grow better in colder climates. It doesn't say anything in here about temperature. So our best answer choice is to protect against herbivores. Number 8. When a stem cell divides, it produces 2 daughter cells. One daughter cell will remain a stem cell while the other daughter cell will differentiate into a specialized cell. Which factor will most directly determine what type of specialized cell will be produced? So when we think about determining anything, we've got to think about where the information lies. So what's going to determine the type of cell? The size, the length of the cell cycle, the number of chromosomes that are replicated, or the genes that are expressed. So when it comes to everything, the way that the genetic code is interpreted or translated is through the the work of the messenger RNA and the transfer RNA, translating the message from the genes in the DNA. So it's all about gene expression. So that information lies in the DNA. It is transcribed into messenger RNA and then translated into protein at the ribosomes. And when it comes to replication is all or none, 46 or none. Number 9. Blue flying fish live in the upper layers of the ocean. Blue flying fish have enlarged pectoral fins that enable them to glide for 400 meters above the ocean surface and appear to fly. Which statement best describes how natural selection led to enlarged pectoral fins and blue flying fish? So, we're going to look at evolution this week, and natural selection is sometimes referred to as survival of the fittest. So, any trait that helps an organism survive and grow up to have offspring and pass on those genes for those same successful traits, those traits will be abundant in the population. Those traits that do not allow the organism to grow and pass on its genes will be uh, rare in the population. So, what is it about these enlarged pectoral fins and the blue flying fish? It's got to be something that makes them successful. The fish were able to glide out of the ocean and into other bodies of water to mate with other species. Well, we know species, you can't mate with other species. You get the sterile hybrids. Fish were able to live both on land and in water as a result of their larger pectoral fins. Well, all it tells us is that they glide above the ocean surface. Doesn't say anything about land. Fish with enlarged pectoral fins were able to glide out of the ocean to escape predators, increasing their fitness. Well, they did glide on the surface of the ocean and appear to fly, so maybe that's it. Or fish changed their DNA to express enlarged pectoral fins, changing the allele frequency of the population. Mutations that lead to changes in the DNA is not something that is willed into existence. So it is something that is passed on to the next generation. So the best answer to this one is got to have something to do with the reason why it's successful when it gets out of the water. It's probably because something was going to eat it. And the last one, number 10, scientists determined the total mass of DNA from a sample of animal cells. The masses of equal numbers of cells were taken during 3 different stages of the cell cycle. Stage 1 contains cells mass during whatever. Okay. So, the mass of DNA during the cell cycle, we have stage 1, 2, and 3. And our dependent variable there is the mass of DNA, and we're going from 0 to 3. So which statement best describes the cells during stage 2? Well, it definitely looks like the amount of DNA or the mass of DNA is the same in stages 1 and 3, but in stage 2, it looks to be much more. So, what could be true about stage 2? The cells have replicated their DNA, but have not completed mitosis or meiosis. So, if they've replicated their DNA, then we would expect there to be more DNA. And if they haven't completed mitosis or cytokinesis, then they haven't divided that. Like with humans, dividing it to 20 to 46 and 46. The cells have completed mitosis and cytokinesis without replicating their DNA. Well, that's not going to work. And if they didn't replicate their DNA, then why did it double? The cells have replicated their DNA and completed mitosis and cytokinesis. Well, again, if you replicate your DNA, 46 times 2 gives us 92 pieces of DNA. And then in mitosis, you would have that. So again, the number would not be double. It would be the same as 1 and 3. And then the cells have temporarily exited the cell cycle without replicating their DNA. Again, if they didn't replicate their DNA, because we're just looking at stage 2, we're talking about stage 2, then the numbers would not be doubled. So the answer is A, the DNA has been replicated during the S phase of interphase. So we have double the amount of DNA. And then we see in stage 3 that mass of DNA drops. And so that's after mitosis, the whole pro meta anatello. and cytokinesis, and we've ended up with two identical daughter cells, and in humans, 46 chromosomes each. Don't forget to submit.